Good afternoon. I'm Jennifer Rose, Chief Commercial Officer of Signios Bio. Today, we are pleased to present Spatial Transcriptomics in KRAS-Driven Lung Cancer. Thank you to Courtney Nurenberchek and our marketing team for producing this webinar. Resistance to therapy remains one of the biggest challenges in KRAS-driven lung cancer. In this webinar, Dr. Mukesh Nyati, professor from the University of Michigan, will share how spatial transcriptomics was used to uncover resistance-associated tumor subpopulations and microenvironment dynamics. Dr. Nyati is a professor in the Department of Radiation Oncology at the University of Michigan Medical School. He joined the department as a postdoctoral fellow in 1999 and has since focused on his research on cancer therapeutics. For over a decade, his work has explored methods to degrade activated EGFR to develop treatments for EGFR-driven drug-resistant cancers in collaboration with Devant Pharma, a University of Michigan spinout that he founded. His research has secured several NIH and DoD grants and multiple international patents. Dr. Nyati contributes to the University of Michigan through roles in the Faculty Senate Assembly, Biomedical Research Council, Rogel Cancer Center Innovation Program, and Carbon Neutrality and Sustainability Council. He also mentors students and organizes the cancer biology course for radiation oncology residents. Outside of academia, Dr. Nyati volunteers for cancer awareness and charitable causes, supporting these efforts through his photography. He'll be joined in the webinar by Dr. Marco Corbo, lead bioinformatics scientist at Signios Bio, who will walk through how spatial data was processed, analyzed, and interpreted. Dr. Corbo is a scientist who has been driven by an interest in the study of genomics, chromosomal evolution, and epigenomics. His main projects have focused on single-cell analysis, bulk and single-cell multiomics, and pipeline development. At Signios Bio, Dr. Corbo is a senior scientist and the director of bioinformatics, where he builds tools and pipelines to analyze large amounts of omics data to aid researchers in drug development and precision medicine. He received his BS in biology from the University of Palermo. Dr. Corbo earned his master's degree in molecular biology and genetics and his PhD from the University of Pavia in Lombardy, Italy. His postdoctoral work was completed at UC Davis, where he focused on identification of mammalian chromosomal chromatin three-dimensional structures and their inheritance in the mammalian phylogenetic tree. I will now turn it over to Dr. Nyati.
Thank you, Jennifer, for introduction and this opportunity to discuss or share some of the work that I am doing along with Signios Bio. Marco has done quite a bit of work that we will share today with you. So our goal is to, I'm trying to look at the next slide, and yeah, I can do it. So yeah, so the goal is to go over some of the introduction slides, and I will talk about a DPI 503, which is a small molecule, and some of its characterization that we have done at University of Michigan or at Devant Pharma. Then I'm going to focus on in vivo efficacy using a few transgenic mouse model. And then we'll raise question about the mechanism of EGFR targeting agent and their efficacy in KRAS mutant model. And after that, I'll just talk about briefly the work that is done by Marco and his group on single nuclear RNA sequencing. And it will followed by Marco presenting the spatial transcriptomics, and then we will have some questions and we'll have some discussion. So this is my one introduction slide, where is actually a lot is packed in this slide. But we know that in epithelial tumors or solid tumors, EGFR is expressed in majority of solid cancer. In fact, it is somewhat difficult to find EGFR null tumors, okay? And if you look at just mutation in KRAS, very well studied oncogene, nearly 1/4 or 25% of all solid malignancy, all solid tumor do have a mutation in KRAS gene. As I said, EGFR is expressed in majority of solid tumor. Therefore, since 1980, people have been trying to develop EGFR tyrosine kinase inhibitors, and there have been about 7 that are approved by FDA, and there are 4 antibodies that are also approved by FDA. So they target EGFR tyrosine kinase activity. But when you look at EGFR molecule, these 11 molecules that have been approved by FDA in USA, their activity is primarily limited to mutant EGFR positive non-small cell lung carcinoma, and maybe in KRAS wild type, BRAF wild type colorectal cancer or head and neck cancer using antibodies. When you look at the prevalence of EGFR mutation, it's only 1 to 2% of total solid tumor patient express mutation in EGFR kinase domain. And when you look at the KRAS inhibitor, there have been multiple inhibitors developed and being developed. There are about 20 programs targeting mutant KRAS, specifically at G12C. Their activity is limited to a site-specific mutation. And with EGFR kinase inhibitor, another issue is that patients do respond when they have mutation in EGFR in the lung cancer, but then almost all patients develop resistance. It's just a matter of time. So we wanted to, in 2008 or 2009, we wanted to develop EGFR degrader. And at that time, the approaches were siRNA, shRNA, etc. Later on, the PROTEC are becoming popular now. But I wanted to develop a small molecule that selectively can degrade activated EGFR molecule. And it took me about 10 to 12 years. And this is where I'm going to describe in next 10 minutes what we have developed. And why we have developed it, this particular molecule or the series of molecules is because of lots of publication from 2008 onwards, only in the preclinical stage or stages, that have shown that if you remove EGFR using either CRISPR, shRNA, siRNA, or transgenic approaches, then whether tumors, whether they are in pancreas or in lung, whether they are driven by G12D or G12C, they all respond to EGFR targeted or targeting agent. So this much we know, that EGFR tyrosine kinase activity alone is not sufficient, or inhibition of EGFR tyrosine kinase activity is not sufficient to produce effect in mutant KRAS tumor, but EGFR removal or degradation is sufficient. So my quest is twofold: one, to develop this molecule and take it to clinic, and the part that we are discussing today is the mechanism. What happens after EGFR degradation that leads to effect in mutant KRAS-driven tumor, because that's a very important aspect. So I want to show you two published papers here. This is first paper. It was published in 2018 in the Science Translational Medicine. And just bear with me, but it's very important finding. That's why it sets the stage beautifully. Here there are two transgenic mouse models were used. Both have lung cancer because of G12D KRAS, where EGFR is wild type. So it's only KRAS mutation or KRAS along with the deletion in P53. What you see is if you have a wild type EGFR, this is the blue line, is the survival. If you knock down one allele of EGFR, you extend survival by 12%. If you knock down both the alleles of EGFR, then you get 40% enhancement in the survival. In the P53 KRAS mutant mice, very similar data were obtained. If you knock down one allele of EGFR, you get 13% survival, which may be insignificant in some way. And if you knock down both, it shows 20% survival. Using the same paper, another paper, people have shown if you use EGFR tyrosine kinase inhibitor, you don't get this survival advantage. Rest is here. You can see that when EGFR is deleted, tumor genesis is affected, and you can see the effect is significant. The second paper is on the pancreatic cancer model, because the mechanism is not limited to lung cancer model, where what has been shown is when you have a mutation in KRAS, it causes metabolic stress because KRAS is always on. So cell must divide. And there is the metabolic stress specifically related to mitochondria, where the mitochondrial ROS, reactive oxygen species levels are very high. And they show the mechanism in which the mROS end up activating EGFR transcriptional expression as well as the ligand. So EGFR is activated as well as EGFR is overexpressed. What is shown here is in these two panels, if you don't have a mutation in KRAS, you don't see EGFR, but when you make mutation in KRAS, you can see the red color is marked by EGFR protein. Later on this article, they also show if you block the mitochondrial ROS activity using MitoQ, then you reduce the stress, you reduce EGFR signaling, you reduce EGFR expression, and you block pancreatic cancer formation. So this is the stage I wanted to set up. And then this slide actually is my about 10 years of work. But as you know, the genomic approach can work, siRNA to protect, but delivering it in patient is very hard. So I wanted to develop a small molecule. So what we did is we began by modeling how EGFR protein is stabilized. And we found that activated EGFR must form a dimer in order to survive. Otherwise, activated EGFR undergoes ubiquitin-mediated degradation. So we mapped the EGFR dimerization site, and we developed molecule based on the structural activity relationship. About 200 molecules were developed to identify DPI-503, which is our lead molecule. It's currently undergoing IND-enabling talks. I want to show you just two-part data. One is in cell-free model, where we just make a purified EGFR protein in the insect cells. And here shown is the activity of DPI-503. This is the structure, where it is radio level, carbon-11. And if you incubate the carbon-11 radioliganded DPI-503 with EGFR that is found in patient, where you have activating mutation, we see a nice titration, meaning as you increase the concentration of DPI, you get a nice activity bound with EGFR. But if you mutate the site, which we model here, we don't get activity. Rest is the cellular thermal stability data, where we compare DPI-503 with osimertinib, which is one of the approved TKI for lung cancer patient. And what you're looking here is, this is the EGFR loaded, just the cytosolic domain. When you subject this to heat-mediated inactivation, it gets aggravated, meaning it gets lost. But in presence of molecule that would bind with EGFR, you actually see that EGFR protein is not aggregated. So this shows a nice binding of DPI-503 with wild-type EGFR, which is very similar to osimertinib. The most important points are in the bottom two, where we show LTC EGFR mutation, where osimertinib binds with this cysteine 797 residue. So osimertinib does not bind with this EGFR, and therefore you don't see heat-mediated. Essentially, you see that protein is degraded or aggregated when you apply 62 degrees Celsius temperature to this soluble fraction of EGFR. But DPI-503 actually prevents this heat-mediated aggregation. Moving forward, wanted to show you that we have in whole cell, this is the EGFR dimer formation upon EGF stimulation, and that is inactivated or inhibited by even 1 micromole DPI-503 in the intact cells, and that causes complete loss of EGFR within 12 hours to 18 hours. And this loss of EGFR is actually selected or selective because we have done proteomic screen where we see 99.6 or 7% proteins are not degraded. Want to show you two on-target activity in vivo. One is using this osimertinib-resistant lung cancer xenograft or xenograft model, where tumors are grown to a very large volume. And then when these animals are treated with DPI-503, the tumors begin to shrink. And you can see within 3 days, EGFR protein is reduced. And then I want to show you the second piece of data. Sorry, my slide moved a bit faster. This is a in-house transgenic mouse model where you have 3 mutations in EGFR in the lungs. So these animals actually develop large tumors within lung. And these tumors are resistant to osimertinib as expected, but DPI-503 does not allow the tumor to really form or grow rapidly. This is where I want to switch the gear. This is the main part of my talk today. So as I mentioned, that those 2 papers, and there are a few more, that shows that if you knock down EGFR, then even when you have a KRAS mutation, whether they are pancreatic, colorectal, lung, head and neck, it doesn't matter, you get activity. So what I'm showing you here is 2-cell line, that is colorectal line, HCT116 or lower. These 2 lines are resistant to cetuximab, which is shown in the blue, and the cetuximab concentration was very high, but DPI-503 is shown in the blue. So in culture, where you have a resistant cell line to KRAS, DPI-503 is very effective. Similar data was shown or found in the UMSCC74B tumor model, where G12D mutation is there in the KRAS, and you see cetuximab does not work, and DPI-503 produce cytostasis. And in another colorectal model, another xenograft model, where DPI 503 has limited activity as a single agent, but when you combine with radiotherapy, we have a very nice activity. This work was funded by Moonshot program. This one slide is actually result from 3 lung transgenic mouse model, where tumors are formed because of either G12D mutation in KRAS, or along with we have FBXW7, which is removed, floxed. This is also another oncogene, and found in about 3 to 4% lung cancer patient. Or when P53 is mutated along with KRAS, what happens with DPI 503 treatment is, you can see here, treatment is actually only for 4-week period. And after 4-week period, we look at the survival. So within just 4-week treatment, which is actually somewhere here, you get close to 60% survival advantage. Now, if you recall the slide that I showed a few minutes ago, where we get about 12 to 40% survival advantage, we have a slightly better or significantly improved survival response in the same tumor model. And in fact, the treatment period is actually very short, and this particular molecule does not cause any toxicity. So when you look at the histology, this is one of the mouse. You can see all the 5 lobes of the lung in mouse, and the darker color shows actually larger tumor volume or tumor area. And this is the DPI 503 treated mouse. You can see the tumors are there, but they are smaller. When you compare the area of the tumor over area of the normal lung, this is what we see is a significant response. And this data is again matches with the published information, where genomic loss of EGFR produce kind of similar trend. So showing that we have a biochemical way or pharmacological way to produce the efficacy that is shown by genomic approaches. Now, the most important question is, what is the mechanism? There are a lot of things have been shown, but we started with single cell or single nucleus RNA sequencing in collaboration with Signios. And then this data, we confirmed some of the findings using cytokine array, histopathology, or biochemical analysis. But then I'll show some of the data and talk about some limitation, and this is where Marco will talk about a spatial transcriptomics. So this in this experiment, we use 2 different mouse model. We use a 2 sample per mouse, so it's essentially a pretty large experiment. This is just shown some of the summary of the data, where what we found in this particular experiment is that 3 cell population, which is endothelial, macrophages, or including epithelial cell population, were quite significantly altered upon DPI 503 treatment. And this volcano chart shows that several pathways like involved in the retinoic acid, steroid hormone metabolism, specific mention of pentose phosphate pathway, fatty acid oxidation, and glycolysis pathway were significantly altered in all both the tumor model. Further transcriptomic analysis shows a significant metabolic shift, a change in the 1 carbon and also energy metabolism and lipid peroxidation. We also saw immune profiling was dramatically altered, and so was the ferroptosis marker. All of these things were actually shown in about 6 other publications where EGFR was knocked out or knocked down using genomic approaches. So it looks like we actually got to the right mechanism for DPI 503. But the limitation, so I'll talk about limitation in a minute, but just wanted to share 3 slides how we confirm the data. So we ran a cytokine array, and we found some of the exact same molecular profiling that we found in the single nucleus transcriptomic analysis were also changed. We confirmed using IHC in 3 tumor model, where we have at least 8 mouse per treatment group, 8 mouse control, 8 treatment group. And you can see that EGFR protein is significantly decreased without affecting mutant KRAS, because that is not our target. And CD31, which marks endothelial cell, is affected, reduced. The mouse macrophage 480, which is a marker, were also reduced. And we saw the CD4 positive cytotoxic T cells were accumulated in, even though the tumors were very small. And we saw the sign of ferroptosis, and which we linked with the change in XCT or SGLT. These are the 2 very important nutrient transporter that people have shown in past, that EGFR protein stabilize physically these protein, and when EGFR is lost, they are affected as well. And the third is the biochemical assay, because other people have shown that when XCT or SGLT are affected as a function of reduction in EGFR within 2 hours with DPI 503, we see nutrient import is affected. That leads to decrease in glutathione synthesis, and therefore we see accumulation of free radical, which is marked by H2O2 or hydrogen peroxide. So we can rescue DPI 503 mediated cell death by giving extra glutamine or extra glucose. And then if you quench free radical, which is induced here by N-acetylcysteine, then we reduce the cell death dramatically, indicating that we have on-target activity. Now, this is all very good, and we are, I guess, in process of fine-tuning and repeating some more experiment and publishing. But then we had one major issue. So first of all, these findings that I discussed are reproducible. But one we had major issue is that in the lungs that I showed a few slides ago, I'll go back to this for 1 second, so that becomes very important, why we did this experiment. When you look at the lung, you have the tumor and you have normal cells here. So distinguishing between epithelial cells, which are tumor versus non-tumor, within the same mouse background is not very easy using single nucleus RNA sequencing. So therefore, I was suggested that we should do a spatial transcriptomic to really understand what happens in tumor cell versus what happens to the neighbor cells to the tumor, and how exactly what transcriptomic profile is altered within different tumor types that we have. And this is where I think I prefer that Marco can talk about it. And then we'll come back to this.
Thank you so much, Mukesh. Yeah, it's been like a great collaboration so far, and then trying to help through our platform and solution the project that you've been working on. We've been pretty excited on this collaboration and all the results that we are uncovering using all these different technologies. And then, of course, trying to stay always on top of the game by using state-of-the-art sequencing technologies and experiments is probably the key nowadays to really dig into the molecular details on how these pathways they play and what's their role within this type of kind of mutation in cancer. For this reason, with Dr. Nyati, we moved forward on proceeding on a different kind of approach, which was a more comprehensive approach respective to the classical transcriptomics, because we wanted to understand how the environment around the cancer cells, and specifically the tumor environment, was being affected not only by these treatments, but also by the genetic mutations that all these models they carry. And for these reasons, we took advantage of the so-called spatial transcriptomics to add another layer of information to the previous molecular transcriptomic data coming from both in single nuclei sequencing to add the spatial context to the cell molecular mechanism and their influence by the different treatments. And here, probably you guys have been seeing this picture. It's pretty popular, but it's a very good depiction on how spatial transcriptomics works, and especially in the context of comparing it to the classical way of sequencing of gene expression. When we think about what it looks like to have a clear picture of what's going on at the level of gene expression, the whole organ. In fact, as you can see on top, this is a representation of how a tissue, in this case an organ, a brain, looks like in a 3D context. And you have so many details on it. You have the colors will resemble the different cell types. You have the stacking up of the small pieces, which resemble the three-dimensional component of the brain. And all this gives you an idea that biology doesn't work by only specific compartments. He works as an holistic, whole comprehensive system where all the pieces, they have an individual function, but also when associated to different tissues and when associated to different spatial contexts, they can add top layers of different function as well to provide the more comprehensive and complex reaction that all the different organs provide. From the classical point of view, we were able to get this information, at least the gene transcription, from a bulk perspective, where all the organ was meshed up, or part of the tissues were meshed up to a single cell level, but then also the single cell level was disrupted through all the prep that the classical method of gene expression was providing. So at the end, while sequencing bulk transcriptomics, you have an idea of what is a mix of this organ and what is the expression, the average expression of all the genes within this organ, but you really don't know what is the actual measurement of every single piece coming out of this tissue. The next level of technology was, of course, single cell and single nuclei, as mentioned by Dr. Nyati, where you can decompose the tissue and the organ, and you can have information from the at single cell level, in this case described by different colors of the small pieces of the brain, and that's a good level of definition and resolution of the data. But still, of course, you only have one information, which is tell me what is the cell specific expression of these genes for each individual cell population, which has been a great frontier of new data coming from all these different publications in the past 10 years. But of course, it still has some limitation in giving the real background on how these cells interact with each other, and then where are they present in the physical location within the context of the organ. And that's very important because, as I mentioned before, cells, they don't work by themselves. They work in a biological three-dimensional space, and they are influenced by other cells surrounding them. And also they influence other cells by segregating different molecules, compounds that react and cross-react with other cells. So the next step was obviously understanding how to put together these pieces in the context of spatial conformation, so we could have a better representation of where these cells are together still with the information of their gene expression. And that's where all these spatial technologies, they're coming in to help. They combine both the histological section, classical from cytogenetic work, with the more advanced techniques of sequencing of gene expression from next generation sequencing techniques, to give you an idea on where cells are present in the specific location of the tissue section, but also what is their gene expression in the context of the spatial representation. Still, of course, if you look at the comparison between spatial RNA sequencing and the actual three-dimensional representation of the organ, we are missing one layer. We are missing another dimension, which is the depth and the third dimension that we are missing from this specific technology. But I'm pretty sure that with the newer technologies that are coming up, we will get that information stacking up on top of the spatial. However, for the moment, spatial has been very helpful on uncovering some of the most important questions that they've been there associated to cancer biology, to developmental biology, disease biology, where the information of the molecular pathways through gene expression is not enough. But coupling that to the spatial localization of those cells was a really important data to obtain. And I want to give you a perspective also on how this technology works. In this case, for the context of this specific collaboration, we use spatial transcriptomics that was created by 10x Technologies. And here, in summary, there is an overall representation of all the different components that the sample go through to provide you the final data. So in this case, we have a section of the tissue of the interest that we want to study. In the case of Dr. Nyati, was a section of lung tissues that we stain through H&E staining. And then here we already have one information that is going to be coupled to the second layer of information, which is the spatial information and localization of the cells. Of course, through a histological section, you can understand where cells are and how they are related to each other in a sense of a spatial context, but you still miss, of course, the information of the gene expression. And because of the technology, we are able to then grab this tissue of interest from a slide and to hybridize it with probes that they are binding to RNA transcript for each individual cells with a specific detail on the read. The probes will have not only the information coming from which gene is being transcribed, specifically where the gene is binding respective to the probe, the UMI tag for correction of PCR amplification, but also most important, the oligos, these probes will have also another tag, which is the tag that is going to be used to recompose the three-dimensional information. So whenever we hybridize this probe to the tissue, each probe will have the the spatial oligo, which will be useful to then recompose the data back again to understand where the data is coming from respect to the section. And here, that's the two final data that we are able to recover. One is just the representation and the organization of in this case, spots from the spatial analysis, which is coming in this case only from the transcriptomics data. Here is a UMAP representation in which all the different colors resemble different cell types. But then, because remember we had that spatial oligo together, hybridized together with the gene expression, we are able to overlay then this information, these all colors, which are the information of the gene expression from each cell type, back again to the original tissue. There was a section on the slide to overlay the gene expression and really understand what kind of cells we were able to retrieve from this specific section. And you can use spatial transcriptomics in so many different applications. People, they've been using in different contexts, in the context of tumor heterogeneity, in disease microenvironment, disease pathogenesis. And you can do an array of stacking up of different layers of information with spatial. For example, you can spike in probes if you're looking for CRISPR-Cas specific gene edits. You can create probe that will target any other kind of biological material in the forms of RNA present or also DNA present in the tissue itself to really uncover some of the information that is being present within the specific tissue type. And here I'm going to show you what you can actually do with spatial transcriptomics and why so powerful. So here there is a characterization of two different samples. For the moment, I'll tell you what specific models are, but these are actual samples processed by Signios Bio that were provided by Dr. Nyati in two of those cancer models. And here I want to summarize the fact that with this technology, you're really able to allow the characterization of tumor microenvironment, which is one of the most important characteristics that you can study within a context of a cancer type. And through the different colors, you can see the different identification of cell types, going from cancer cells to macrophages to other immune cells. And not only the identification of the individual cells, but also where they are physically present within the spatial context of the tissue, which makes at the end what a tumor microenvironment is composed of. And then through a series of techniques, which includes marker-based cell identification and overlaying with other external datasets, we are able to dig into the tumor microenvironment composition of the different cell types, starting, of course, with the cancer cells, because as you remember, one of the main challenge from the previous experiments that Dr. Nyati showed is we are not really able to recognize and dissect cancer cells and distinguish them from other epithelial or endothelial cells through classical means, even including single nuclei, although with the newer technologies and newer tools, now the identification is becoming a little bit better. But of course here you can distinguish not only those cancer cells because you can overlap the gene expression markers together with the staining of those cancer cells, and you can get the identification of the cell types, but also where they are physically present in the context of this tissue. And then following of that, of course, one of the other important point is, what about the other immune cells, and how do they play a role compared to the cancer cells with respect to my model and to my treatment? And this is another example of identification of another cell type, in this case macrophages. And in this case, as you observe compared to the previous slide, macrophages, they are surrounding the cancer cells in this specific context, while here they form some of the hotspots of recruitment for these specific cell types. And then we go to another layer of the tumor microenvironment, specifically to another cell component of the tissue, which is B cells. Through specific, again, markers and data set, we were able to identify those cells which are way more prominent in the tissue. They are very sparse, and they're still overlapping the surrounding area of where the cancer cells were identified. And then when you overlay all this information together, that's where the context of the spatial characterization of the tumor microenvironment really gives the best power out of the current technologies, because you can see, you can overlay, as I mentioned, the histological information of having an understanding of where, from a classical means, the cancer cells were present, by overlaying to a more next generation sequencing approach, by sequencing the gene expression of those cells. You can really compare then localization of those cells to their specific expression, and then dissecting all the different cell kind and what is their relationship towards each other, but also towards the model in which that we are trying to understand, and also what are the consequences of the specific cell types after we induce a treatment process with some of the molecules that Dr. Nyati has been showing. And that's definitely one of the most important representation of what is the power of spatial transcriptomics. And of course, you can do a lot of downstream application, because from here, this is just the start of the data. You can open up this data for so much downstream application and start asking a lot of questions. For example, we want to understand whether we're looking at two different models. In this case, it's the KRAS P53 mutation, where D stands for the treated sample, and B stands for the vehicle, so for the control. We want to understand how cancer cells they are differentiating and they are being affected in the same model by the treatment. And you can ask what is the colocalization of the gene expression together with the specific cell kind, and starting, for example, by asking, okay, tell me something about some housekeeping genes. So when we check the ACTB expression, you can see there is a higher expression of the surrounding cells respect to the cancer cells, here represented with the darker blue. And then when we ask about the expression of some markers, they are known to be more expressed in cancer cells in the specific context of lung cancer, where they are expressed, and if they are expressed at the same level within the two models. But what we see here, for example, is one of the markers, PBA, is being expressed more abundantly in cancer cells in both models, both the treated one and the untreated. We see that in some case, cancer cells, they don't behave exactly the same. In this case, for example, the expression of SOX2 is different when we compare the same cell type after and before treatment. Here, we see that this cancer, they still express, say, the rescue or the expression of this component, while it's a little bit more turned off in the vehicle group. And that's one of the other important data that you can get and you generate out of this technology, because you can also see the location of where the cells they are expressing the specific marker, and whether the marker is being surrounded by other cells, let's say immune cells in the context of the tumor microenvironment, to see whether those cells they are influenced by surrounding cells, or they behave independently just based on the treatment alone. And to just give you a little bit more data and trying to understand whether the spatial information could be a further proof of what Dr. Nyati has been showing as results to other different technologies and techniques like the single nuclei. We look at the same pathways and some of the genes, and this work, of course, is still ongoing, but I'm showing some of the preliminary results. So in this case, what I'm showing is the expression level of key genes that they are components for pathways of nucleotide metabolism, glycolysis, and TCA cycle, to look whether there is a treatment-specific change. In this case, we are comparing the KRAS positive, P53 positive model. On the left, we have the control, the vehicle. On the right, we have the DPI-induced and treated cells. And you can see when we look at the different key markers, they are expressed in a different way, not only within the same model, where different cell types express these markers in different ways, also when compared to the two different models at the same cellular resolution. You can see, for example, for the glycolysis, genes like exokinases HK1, 2, and 3, they are more expressed in the treated group compared to the vehicle, and especially in macrophages, alveolar cells, and endothelial cells. And what you observe is we are able to confirm some of the findings shown before, that this is also the case for other pathways. They show upregulation in the treatment compared to the vehicle. For example, when you look at the nucleotide metabolism, most of the cell type, they show a higher expression of this marker compared to the vehicle itself, including the key genes for the TCA cycle. We were able to see some of the opposite representation of gene expression. In this case, when we look at the TKT, we see a higher expression in T cells in the vehicle when compared to the control. And then to overlay this information with the spatial analysis, here we are asking the same question, but with the different techniques to the data. Specifically, we are asking, okay, tell me where some of the genes they are expressed, and I want to know whether they are more expressed in the treatment compared to the vehicle, or vice versa, just by keeping still the three-dimensional the spatial information of the gene expression. So in this case, for the specific exokinases 1, you can see that the expression is modulated and affected not only by treatment, by the model itself. When we look at the KRAS only and the KRAS plus 53, you can see we have an increased expression of this marker in the treated model, both for P53 and KRAS only. And the overall expression is also, as shown before by the violin plot, it's ubiquitous and present in most of the cell types. But then when we look at the Flox F3B-XY7 model, we see that the pattern is slightly different. And then although an overall decrease of the expression of this marker, we see that the difference between treatment and vehicle is not as big as we observe in the other two models. And then when we're looking at another key gene for energy production in the glycolysis pathway, specifically FBB2, we see the expression in this case is not very relevant from the treatment perspective, but we see a huge difference between models. In fact, we observe almost no to low expression in the P53, a mild and low expression in the Flox model. But then in the KRAS-only model, you can observe that this gene is particularly highly expressed in cancer cells. And if you remember from the previous slides, this block here and this block there highlights and localize cancer cells. So they are very rich for this marker all in this model. And not only that, but also that there is a big difference between the treatment model compared to the vehicle, where cancer cells they are overexpressing this marker. And then another important also data that we can get out of this experiment is, as you remember, there were other hotspots of cancer cells in the same context, in the same tissue, which do not overexpress this FBP2. And this is another powerful information we're getting from the spatial transcriptomics, which is we are able to recognize subsets of cancer cells even within the same model, which is very important to uncover from a more complete perspective what is the tumor microenvironment, because we know that different cancer cell types might be at different stages within the same model, so you can tackle and understand better in using these technologies. So in conclusion, from this experiment that is still ongoing, so these were like preliminary results, Dr. Nyati showed that DPI 503 treatments results in a substantial metabolic reprogramming in lung cancer cells by enhancing glycolysis, oxidative phosphorylation, and altering fatty acid metabolism, but also decrease the pentose phosphate activities, which leads to an improved survival rate in preclinical models to reduce tumor proliferation. And also by using spatial transcriptomics, we show that tumors, despite looking similar, have diverse molecular signature, and then also they relieve significant tumor heterogeneity by identifying not only same cell types expressing different level of markers, but also identifying from the same cancer cells subgroups of model-specific cancer types. In addition, the DPI 503 treatment is caused distinct molecular changes in different tumor areas that Dr. Nyati, together with our collaboration, we are trying to help to identify resistance to sensitive tumor profiles. And also at the end, Dr. Nyati was able to find that higher ferroptosis marker suggests a new way of how DPI 503 suppresses tumor than traditional TKIs. And overall, after the conclusion, this is where the study is now moving forward. Dr. Nyati is planning to add a new model to the pipeline together with some pancreatic model. We're gonna do a deeper dive into the spatial analysis, because the next step will be the characterization of the surrounding area of the cancer cells and their interaction with the immune cells, adding some metabolomic study, and also define the mechanism of action of the in more molecular details of DPI 503. I hope there was a good overview, not only of the technology, but especially how the technology can help really facilitate the study of the mechanism of such important models. And that's where I want to thank Dr. Nyati for this collaboration and for trusting us with this work. And I'll open the stage for questions, and thank you.
Thank you, Dr. Nyati. Thank you, Dr. Corbo. We now open the floor for questions. You can use the chat box, the chat functionality within Zoom to send over a question. Give everybody a minute. There's a couple questions that are coming in. The first one is for Dr. Nyati. What is the main hypothesis on how EGFR degradation mechanistically leads to in vivo tumor regression in KRAS models?
And that is a question we want to fully understand and address. But what has been shown previously and what we confirm data, which I showed briefly today, they do align, where what we see is actually shown in this diagram, at least to some extent, that when you block EGFR tyrosine kinase activity shown in the second receptor here, it does inhibit EGFR signaling in the event when EGFR is activated within the kinase domain due to mutations. But even the inactivated molecule here, which is shown in the gray color, is very important in producing survival signaling, because it does work like a molecular chaperone, where when receptor is inactive, it stays on the plasma membrane and binds with many things, including KRAS indirectly, and binds with these receptor like SGLT, XCT, and many other, and allows them to function properly to take the nutrient balance or import properly. When you remove EGFR, then these molecules, like two of them at least we confirm, XCT and SGLT, they are significantly altered, and therefore that causes stress, metabolic stress, and because of the lower level of glucose and lower level of glutamine, cell cannot synthesize enough glutathione 6 peroxidase, and because of the mutation in KRAS, cell continue to still proliferate. That causes mitotic stress, which is referred as mROS. And when you have a higher level of mROS and lower level of glucose and cysteine, that state is not very, that is actually vulnerable to mitotic stress, metabolic stress, and cell death. And this is how other people have shown, and our data clearly indicate that we can reverse it by multiple stages, by just giving more cysteine, more glutamine, more glucose, quenching the reactive oxygen species of free radical. We can reverse the death that is induced by 503. So this is what we think is one of the effect on the tumor cell. But then we are also figuring out or finding what Marco showed today, how the tumor microenvironment is rewired. When you look at some of the diagram, and obviously cannot discuss all the detail yet, but when you look at the T cell versus endothelial cell versus epithelial cell, tumor cell, these changes are differently occurring upon loss of EGFR, because EGFR is more important in tumor cell than many other cell types. So how exactly cell-cell interaction effects we don't fully know yet, but this is how we like to believe that mitotic stress induced by loss of EGFR protein causes metabolic stress that puts cells into the vulnerable situation that causes cell death by itself. And specifically when we combine with low dose of radiotherapy or cisplatin, which is also causes DNA damage, then we get a synergistic effect. At the same time, when we use cetuximab, which is the antibody that blocks EGFR kinase activity but does not induce degradation, we don't see this effect. So we are currently working on identifying specific PROTAC and siRNA and shRNA to compare these studies along with DPI 503 to really nail it down. It'll take some time to say one or two things. Right now, it's a little bit more descriptive.
Thank you, Dr. Nyati. A couple more questions have come in. There's a question to Dr. Corbo. What exactly do you need for spatial transcriptomics analysis service? How many slides or the size of the tissue block? And are there any special requirements for tissue prep?
Thank you, Jennifer. Thank you for the question. So for the technology, in this case, we are talking about the 10x Visium spatial transcriptomics. There are two main versions of the technology. One is the ones that we show here, which has spots bigger than a specific cell type. So you won't really get the single cell resolution because you're going to have around 5 to 10 cells within the same spot. And then there is another version that is called the HD, which has 8 micron beams. So it's more closer to single cell resolution. And for those both technologies, you can use fresh frozen and FFP slides. And the capture area that you can work on to do the spatial transcriptomics is of two kinds. One is a 11 mm by 11 mm, and for the Visium HD, the capture area is 6.5 mm. So depending on what you're trying to look at, you can decide one or the other, and then you can have multiple capture areas in the same slides, up to 4 samples. So you can either do replicates or different serial section of the same sample in multiple capture areas, or you can use different samples in the same slide in different capture areas. But also, if your tissue type, let's say it's smaller than 11 mm, you can put together more tissues to enclose in the same capture area, so to take advantage of the whole space. Then you're gonna send the block, or you can send the curls. And then I can connect you with our technical lab team for the details of how to send those. But then you will gonna send the block, and we can do the staining and then the post-processing about cutting from the curls from the block, and then putting on the slide and then proceeding into the tissue prep.
Thank you, Dr. Corbo. And we're running out of time, so we have time for one more question, and this is actually to both Dr. Nyati and Dr. Corbo. Do you foresee spatial transcriptomics guiding therapeutic targeting in KRAS mutant cancers? Both can take a turn.
That is certainly the hope. That's because what Marco also mentioned, one very important finding is that within the same lung, the same mouse, that is, 2 tumors, because there are hundreds of tumors, 2 tumors are not same. And it probably because of what else is growing around the tumor, or what is the volume of the tumor, that changes its transcriptional profile, immune profile, etc. And what we are also finding that DPI treatment is also not, the response is also not homogeneous across the different model or within the same model between 2 tumors. So the hope is that we really want to understand transcriptomics, metabolomics, and immune profile of tumor that responded better to this agent versus that didn't respond. And if such information can be dialed down to 2 or 3 transcript or protein, then the hope is that we can at least we don't know whether we can translate, but hope is whether we can translate that information to identify a patient who would likely to respond or not respond to a particular series or class of therapy. So yes, the answer is certainly very optimistic, but we are not there yet.
Yeah, definitely.
Dr. Corbo.
Yeah, I would say that Dr. Nyati really fully exposed what is the picture of the situation. Something extra that I will add is nowadays with all the other omics assay, proteomics, metabolomics, and genomics itself, you have an extra layer of information that nowadays with all these machine learning AI-based tools, you can stack up layer on top of each other to add as a, let's say, a multiplex information experiment. So to couple the spatial with other different kind of projects and solutions to have more information of how the spatial context of the tumor is related to also their metabolomics and the proteomics. So that's very exciting, and all this is being done nowadays. But definitely one of the challenges, the overlay of such different diverse type of data sets is very challenging, not only from a computational point of view, but also from a normalization and standardization. So that's one of the main challenge, but also one of the exciting advancements that is ongoing on staking up multiomics techniques.
Agree with that.
Thank you, Dr. Corbo, and thank you, Dr. Nyati and Dr. Corbo, for the presentations today. We are at the end of our time, and this concludes our webinar. We will send out a recording, and thank you very much.
Thank you.
Thank you. Thank you, Jennifer. Bye-bye, everyone.